Bom dia a todas e todos que estão aqui na sala do Conselho Universitário e que estão acompanhando a transmissão online. Eu sou Paulo Nussenzweig, Pró-Reitor de Pesquisa e Inovação da USP, e a gente tem hoje mais uma USP Lecture, essa série que é tão importante para promover o ambiente acadêmico na nossa universidade. O nosso palestrante de hoje vai ser apresentado pelo professor Rodrigo Calado. Eu não posso deixar de fazer uma menção especial ao palestrante de hoje e tentar me defender de eventuais acusações de nepotismo. Michel Nussenzweig é meu primo. Mais um dessa estranha família de tantas pessoas que se dedicam à ciência. Não é só mais um. O Michel é membro da Academia de Ciências, National Academy of Sciences, dos Estados Unidos, e eu acho que o Rodrigo não ia dizer isso na apresentação formal dele, mas o orientador de doutorado do Michel foi laureado com o Prêmio Nobel, e foi um laureado com o Prêmio Nobel ímpar, não só pela ciência que fez, mas porque faleceu no fim de semana antes do anúncio. Então foi o único até hoje que foi anunciado póstumo. E para imenso orgulho da família, o Michel foi quem foi apresentar a Nobel Lecture do Ralph Steinman. Então eu vou deixar o Rodrigo apresentar a parte científica do Michel e dizer que, especialmente hoje, é um prazer muito grande ter o meu primo aqui.
Bom dia a todos e todas aqui no Conselho Universitário, assim como aqueles que nos veem pela apresentação, pela transmissão direta dessa USP Lecture. Para mim é uma grande honra e um privilégio, nesse momento, poder fazer a apresentação do professor Michel Nussenzweig, que é paulistano, mas ele estudou nos Estados Unidos, onde ele fez medicina na NYU Medical School, e depois doutorado na Rockefeller University. Ele fez residência médica em infectologia no Mass General Hospital, e desde 96 é professor titular de imunologia da Rockefeller University. Ele é pesquisador do Howard Hughes Medical Institute, que é um dos principais órgãos não públicos de fomento à pesquisa na área biomédica nos Estados Unidos e no mundo. Ele já recebeu vários prêmios e distinções científicas, entre elas o prêmio Robert Koch, do governo alemão, dedicado especialmente a pesquisadores na área de infectologia. É membro da Academia Nacional de Ciências dos Estados Unidos e membro correspondente da Academia Brasileira de Ciências. A pesquisa científica do professor Michel Nussenzweig, ela é voltada principalmente para entender os mecanismos imunológicos, tanto da resposta inata quanto adaptativa, contra as infecções, em especial as infecções virais, contra o vírus HIV-1 e, mais recentemente, com a importância que todos sabemos do SARS-CoV2, sobre o qual ele vai falar hoje. O trabalho dele consegue demonstrar como que o linfócito B monta a resposta imunológica e monta a produção de linfócitos frente à infecção, frente ao desafio com essas partículas de antígenos virais. No nível mais específico e profundo, seja nível genético ou molecular. Como, por exemplo, como ocorre a hipermutação nesses linfócitos, o que auxilia no desenvolvimento de vacinas mais efetivas. Por outro lado, esse outro conhecimento me é também bastante importante como hematologista, porque nos ajuda a entender como são desenvolvidos os linfomas de células B e como entender os mecanismos e a biologia dessa grande classe de tumores. Adicionalmente, a sua pesquisa ajuda a entender como as células dendríticas, que fazem parte da resposta inata imunológica e que processam os antígenos dentro do nosso organismo, como que elas podem tanto induzir a intolerância imunológica frente a diversos antígenos, ou, contrariamente, ativar os linfócitos T para montar uma resposta imunológica. Então, com essa breve introdução, gostaria, então, de chamar o professor Nussenzweig, por favor.
Muito obrigado. Eu ainda consigo poucas palavras em português, mas acho que vou ter que falar em inglês, porque o meu vocabulário é de menino de 9 anos e está enferrujado. Okay, so what I'm going to try to do today is I'm going to try to give a brief introduction to what immune systems are, with apologies for people who are really expert. And then I'm going to try to explain something about why there's a disparity between our ability to be infected by, for example, the coronavirus, and our ability to essentially fight it off. So what happened between the time when we were initially challenged with this and now. To begin, I want to just focus on what the properties of the immune system are, and a construct, a theoretical construct, to explain these properties. So the immune system has really 3 things that are different or unique about it. And they are specificity, diversity, and memory. Specificity is something we all know about, which is that when you're challenged with a particular immunogen, you react to that immunogen, but you do not necessarily react to other unrelated things. So that's specificity. Diversity is a property which says that the immune system can see essentially a universe of different immunogens. And this was initially something that was discovered in the beginning of the last century, or sort of the middle maybe, when scientists, actually a scientist at Rockefeller, Carl Landsteiner, made compounds that did not exist in nature and then immunized animals with those compounds, and found that he could raise a specific immune response to those compounds which didn't exist in nature. So that meant that nearly anything that you could use as an immunogen would produce an immune response. So that's diversity. And memory is the property of immune systems that's useful in vaccination, which means that if you are challenged with an immunogen, then a subsequent challenge will lead to a better, faster response. Okay, so how do we explain those different properties? And there's a very simple model that was put forward shortly after actually Landsteiner made his discoveries, which explains these properties. And it's illustrated here. It says, immune systems are built out of cells, and each cell has a unique specific receptor. So specificity comes from the receptor on the cells. And then diversity comes from the fact that there are many, many cells. Each one has a different receptor. So both specificity and diversity are explained by this. And then memory can be explained by the fact that when an immunogen comes into the system, it selects for the cell with the right receptor, and that cell is then expanded into a clone. And if you are re-challenged, you have a bigger clone which can respond faster and better. So this is a very simple way of thinking about immunity, and in fact it's in many respects it's correct. They got the Nobel Prize for this model, essentially. Now there are some things that this model does not explain. and two properties that the model doesn't explain and that are both important for understanding how this coronavirus thing is happening are illustrated here and were discovered in the 60s by this man, Hermann Eisen, which I got to know very well actually, and it was really a wonderful mentor really. So what Hermann discovered was that when he immunized animals with an immunogen, that the affinity of the antibodies in serum increased very rapidly. And that's a very hard thing to explain based on that initial model. The other thing that he discovered, and that was he couldn't understand and had actually been very hard to understand for a long time, is that in addition to increasing affinity, there was also an increase in diversity at the same time. So he was a physical chemist, and it was hard to explain how something could become more higher affinity and also more diverse at the same time. So we've learned a lot about how this process of affinity maturation occurs, and I've tried to illustrate it here. So the first part of the Burnett idea, that there are clones of cells that undergo expansion after antigen challenge, is correct. But something that Burnett did not anticipate, and that we've only learned about fairly recently, is that when cells begin to expand, they express an enzyme, a cytidine deaminase, which does something really strange and unique to this cell type, which is it's a mutator, and it makes mutations, and it makes mutations specifically in antibody genes. So instead of having a monomorphic clone that's expanded, it's really a collection of cells. Each one is a little bit different from the other, and it's different by mutations in antibody genes. And then during the immune response, there is selection for the cell that has the high affinity. Now, this process happens in germinal centers, and my colleagues and I have been studying these things for some time, and this whole process and how it happens, and we really have learned a great deal about how affinity maturation occurs. This structure, it's called a germinal center, it has two products. It produces memory B cells, the cells that stick around and can respond subsequently. And it also produces plasma cells. These are two different cell types, both of which are antibody-producing cells. Plasma cells live in the bone marrow, for example, and are continually putting out antibodies which are in serum, and those are the antibodies that would protect us from being infected. The memory cells are an entirely different compartment, and they are silent. They are long-lasting, circulating cells, but they do not produce the antibodies that they encode genetically. They're just sitting there. Plasma cells are selected exactly this way. So they are high-affinity cells. Memory cells we've known really very little about until quite recently. And that's in part because we had very little in a way of capturing, learning about these cells. So what I'm going to just tell you very briefly about is a mouse experiment that was done in my lab by postdoc Charlotte Viant not very long ago. So instead of capturing memory cells using markers, which is what had been done in the past, she decided that she would look more generally, more broadly, for all memory cells. And to do this, she needed a genetic tool. And the genetic tool in this case is a driver gene, which expresses CRE, which is a recombinase, which allows controlled deletion of a stop codon in front of this ROSA 26, which is allows very specific expression of the gene, a green marker, in germinal center cells. So everything that comes out of germinal centers is permanently marked green, GFP. So now you can look for what is memory. So when she did this, and this is the experiment is you immunize, in this case with an HIV antigen, you turn on the gene with tamoxifen, and now you look in germinal centers and in the memory compartment. So the marker is in this axis, and this is a control. It's not labeled. These guys have germinal centers, so they're labeled. They're further down. And then this is the antigen, the immunogen. So cells in germinal centers are binding to the antigen. That's what this is. And memory cells really look pretty empty. And that was a real surprise, because the expectation was that the memory cells would also bind to the antigen, that they would also be specific. But in fact, there were very few such cells. So, because this is flow cytometry, and we wanted a more physical sort of measurement, we went ahead she went ahead and cloned the antibodies from those two compartments. And this is a BLI experiment which measures affinity. And in this setup, we're looking for a monovalent interaction between the antigen and the antibody, the Fab. And what we saw was that germinal center cells produced antibodies that bound in red, but very, very few of the antibodies in the memory compartment bound. So that was exactly like the flow cytometry, and surprising. But in vivo, these interactions are not monovalent interactions. They're polyvalent interactions. There's a cell that has multiple receptors, and there's another cell that's capturing the antigen, and that the antigen is multivalent on the surface. So we changed the valency by changing the setup so that the antibody became a polymer on the surface of the chip, and now we look at the antigen binding, and now antibodies that did not bind monovalently in the memory compartment bound. And if we further increased the valency, then we could see even further binding. So it wasn't that the memory cells don't bind the antigen. They do bind, but they're very diverse. So the selection mechanism for memory is entirely different from plasma cells. And what comes out in the memory compartment is a real distribution of cells that have these mutations in antibody genes, but aren't necessarily the best cell for the immunogen. On the other hand, they could be a good cell for something related to the immunogen, a mutant, for example. So what the germinal center produces is high affinity plasma cells and a collection, a library, essentially, of memory, which is diverse. All right, so with that as sort of background, I want to tell you a little bit about our work on the coronavirus. And I believe that this background will help explain some of the things, some of the phenomena that we're all experiencing. So our lab has worked on this problem really quite a bit. And what we've done is to look longitudinally at people who were convalescent. In other words, they were infected. And then we sampled them multiple times after the infection. We also vaccinated those people and sampled them after vaccination. We also looked at people who hadn't been infected and were just vaccinated with the mRNA vaccine. We looked at people who received the Johnson vaccine, adenovirus vaccines, and combinations of vaccines, and several other studies which aren't even on here. The tools that we used to study these people, so many labs measured serologic activity. That's very simple. You just take serum, plasma, and you do various measurements. including binding and neutralization and so on. Our focus was a little bit different. We wanted to understand memory. And to do that, we used a tool that we had developed in the lab some years ago for finding B lymphocytes that are antigen specific. The way we do that is to collect blood, purify the cells, and then do something that's really trivial, which is to use the antigen, the immunogen, to bind to the cells, to find the cell that has the specific receptor for that immunogen, and we use flow cytometry to isolate single cells that bind to the immunogen, and then do molecular biology to clone those antibodies. That's turned out to be a very, very useful technology. It's a technology that was used by Lilly and Regeneron and all the other companies to clone antibodies from people and put them back into people. And they knew that that would be safe because we had proven that that was the case in our work for HIV. So this is a very, very rapid method. Potentially takes just a week to do this whole part. And then there's time in order to produce it for commercial use or for putting it back into people. It's much faster, for example, than most vaccines, and it's much faster than making chemical compounds that are safe for people. That's why the antibodies were really the first tools that were used in this pandemic. It's because of this method. All right, so that's what we use to find the lymphocytes and characterize them. And then this technology was developed by a colleague at Rockefeller, Paul Benesch, which is a pseudovirus assay for whether or not you neutralize the virus. And it's based on luminescence and blocking infection. Although it's a pseudovirus assay, it's pretty much equal to a real virus assay, and that's what's illustrated in these two graphs. Now, when we started doing this, and we started very early in the pandemic, one of the first things that we noticed was that people who were infected were making plasma antibodies, so from plasma cells, but that the level of antibody that they were making was really not very strong. So this graph, just each one of these bars is an individual, and this is their neutralizing activity, and these are the first 60 people, and below these 60 people, it was really pretty hard to detect neutralizing activity. So although they had been infected, although they made an immune response, it was not a very strong immune response. Now, we knew a lot about the structure of the protein that is necessary to get into, for the virus to get into cells. That protein is illustrated here. It's the spike. And the part of the spike that actually contacts the cell is this thing here called the receptor binding domain. It binds to this cellular receptor ACE2. And because we knew this, we used this piece of the protein as the bait to find the cells that produce the antibodies that are specific for the virus and that neutralize. One of the things that we found immediately is illustrated here. So these are what a flow cytometry looks like, and these little dots in here are the cells that actually bind the antigen. Each one of these pie charts illustrates, the number in the middle is the number of antibodies that we cloned, and each one pie chart is one person. The slices are the clones. So the clones that Burnett hypothesized are these slices, and the colors are shared antibodies between people. So one of the findings was that people are making very, very similar antibodies, different people. And because we had these antibodies, we could do their physical structures, and that was collaborative with Pamela Bjorkman and Christopher Barnes. And what we found, and what they illustrated with this is that just about everybody, whether they were infected or they were vaccinated, made this class, these two classes of antibodies first. Class 1 and Class 2, they called them. So everybody was making the same thing. And these two antibodies bound exactly to the footprint, the ACE2 footprint on the receptor binding domain. And they covered up these red residues here. The red residues are the residues that were the first mutant escape residues that came up in Brazil, that came up in South Africa, that came up in England. And we could explain potentially why we saw escape, because the whole world was making these antibodies and putting pressure for selection at this location. And you could understand this, or you can actually prove that that's the case, by looking at the structures and then taking in various antibodies illustrated here and asking for whether or not one particular class was susceptible to these mutations. And in fact, Class 1 is susceptible to 417 and 501, which was in the Brazilian variant. And Class 2 was susceptible to 484 mutations, shown here, also in the Brazilian variant. So that explains why when we looked, and everybody else looked at serum from people who had been infected with the original strain for whether they would neutralize, for example, this gamma variant that came up in Brazil, that you saw a decrease in neutralizing activity because the antibodies that everybody was making were targeting residues that were subsequently selected against. And you can prove that the antibodies actually do the selection, that's this experiment, by using a virus that undergoes rapid mutation in vitro and putting the spike protein of the coronavirus into that VSV, and then passaging it in the presence of antibodies. So if there's selection, you would expect to see it in the mutations that come up. And that's exactly what was done here. So in this paper, this in vitro selection experiment predicted exactly the mutations that would come up in the gamma variant here in Brazil. What we learned right away was that there were these classes of antibodies that are abundant, that they're sensitive to these viral mutations that are almost predictable from the structural data, that we could do this in vitro, and that the class 3 antibodies, subsequent class, put less selective pressure on the virus. But as we now know, these less frequent antibodies eventually were able to produce selection and lead to variants that we have around today. All right, so what about the memory compartment, and what about the evolution of immunity in people that were infected? So what we and everybody else learned was that, of course, the immune response, or the plasma antibodies, go down with time. So the plasma cells are produced in a big wave, and many of them die within a short period of time. And because they die within a short period of time, the neutralizing activity in serum, initially represented by this light gray, each dot is an individual, drops, and drops very significantly after 6 months. But between 6 months and a year, this compartment is relatively long-lived, and so the amount of antibody in plasma stays very similar. If these people who were infected are boosted, that's the blue, they are really champions. So they get really good antibody responses, also illustrated here, not just to the Wuhan strain, which they were infected with and boosted with, but to other strains. So that is similar to Hermanisen. That is, there is a increase in affinity and increase in diversity. Now, why did that happen? So this begins to sort of explain some of that. And that's here. So here, each one of these columns is one person. Again, the pie charts are antibodies. Again, the number in the middle is the number of antibodies. And the colors now are shared clones, so the Burnett clones, shared at different time points, 1 month after infection, 6 months, and a year. And what was surprising about this was not the shared persistent clones, but the gray clones, which weren't present at earlier time points. That means that even after a year, people who had been infected were still making new clones of antibodies, so persistent immunity. And that's also illustrated by mutation, which also increases with time for an entire year. Now the antibodies, and each dot is one antibody here, and cloned at different time points, 1 month, 6 months, 1 year. You can see here from the IC50s, that's the neutralizing activity. The number is getting lower, which means they're getting better. Okay, so getting better and better after a year. So these people had not been vaccinated, they'd just been infected, and their antibody responses are getting better and better. And the clones, you can see here, the clones in particular are getting really a lot better, the persistent clones. Now they're getting better for neutralizing activity against Wuhan, but they also got broader. In other words, they were able to deal with mutations. So here, you can see each one of these is a pair. So 1 month and 12 months. And each one of these is a mutation in the original Wuhan. And so this pair is sensitive initially to this mutation 417, a common mutation that came up in the variants. But then after a year, it's good. And also against this 501. And in every single case that we measured pairs, the subsequent piece of the clone after a year was better. And there were examples like this where it was resistant, it didn't get the virus initially, but then every single variant is neutralized. And we believe this happened because even though we clear the virus from our upper respiratory tract, there are pieces of virus or nucleic acids that remain and here in the gut. So there is persistent immune response, probably because there's persistent antigen, persistent immunogen in people for very long periods of time. Okay, so for natural infection, there's prolonged evolution to higher affinity, broader antibodies in memory, not in plasma. So plasma, remember, levels are going down, but memory, which is pretty stable, you have all of this stuff. So that means that when you're challenged, then of course you're going to call on that memory, and you're going to have a rapid response, which is going to clear the virus, so you will be infected, but you won't go to the hospital. Okay, so now what about people that are vaccinated? Well, so people that are vaccinated are a little bit different, and they're different in a really interesting way, actually. They too, after on your third boost, not your second, but your third, make really, pretty good antibodies to variants. So this is Omicron, for example, and this vaccination is with Wuhan. So even though the vaccination is with Wuhan and the boost is with Wuhan, everything Wuhan, you still make some Omicron. It's not enough to protect you, but nevertheless, if you're infected with Omicron, you don't go to the hospital. Okay, so what's going on in the memory compartment here? So here, this is the number of memory cells in these people over time. So there's longitudinal, it's the same person. And each one of these individuals is increasing the number absolute number of memory cells. So you're getting really a very large memory compartment after being boosted the third time. What about the antibodies? Well, the antibodies are also getting better in this cohort. They're getting, you can see the number is getting lower and lower with time. But in this case, it's not the conserved clones that are getting better, right? So the conserved clones are pretty much staying the same. And what's getting better is actually the new clones, the newly appearing clones. And that was it's a bit of a surprise, which you didn't really quite understand. And so we used physical methods to map the antibodies. So remember, this class 1 and class 2 is what comes up first. The red ones here are the Omicron mutations. So you can see how antibodies in these classes, including class 3, are essentially knocked out by Omicron. But after and these pie charts now are showing where the antibodies bind. And you can see an initial dominance of class 1, 2, 3. Then, after the third boost, something different happens. And antibodies are now targeting this region, which is the most conserved region on the receptor binding domain, and the one that is hardest for the virus to make mutations in. How did that happen? Well, when we saw this, what we were thinking is, well, maybe the antibodies that you make first actually block here, so the immune system chases in a different place. And that idea is a really old idea. It was first put forward in 1908 by this man, Theobald Smith, who did these kinds of experiments in animals. And actually, a lot of people studied this phenomenon for many years, around the 60s, including this person right here. That's not me. That's my father. And made significant contributions to understanding this phenomenon, but not so much in humans. And so we actually were very fortunate to be able to do an experiment in humans by mistake that spoke to this problem. And so when we were doing the initial cloning, we had all of these antibodies that were very potent and broad, and we were approached by pharmaceutical companies to take the antibodies to be able to put them back into people for therapies. And one of these companies actually produced our antibodies for people, these two antibodies here, which were very similar to the Regeneron antibodies in their activity. Now this company, Bristol Myers Squibb, was a little bit slow, but in any case they made these antibodies for human use, and we at Rockefeller did the first safety studies to make sure that they were safe for people before being able to give them to the general population. By the time we did this, or shortly after we did this, there were mutant viruses that escaped these two antibodies, so these antibodies were useless, basically. But we didn't know that at the time. So we put them into people. They had very good pharmacokinetic properties, that's what's shown here. And these people were vaccinated subsequently according to CDC guidelines, which is 90 days after they got the antibodies. Okay, so how did they do? Well, they're the green ones here, and this is neutralizing activity. So they had a lot of neutralizing activity. They were better than controls. So we didn't do anything that was bad, that's for sure. If we made mutations in the test virus that wiped out the two antibodies that we put in, then they were a little bit worse after the first vaccine dose in terms of neutralizing activity, but then they caught up after the second dose. So they were fine. But the antibodies they made, we cloned them, were really interesting because instead of the green here, controls, class 1, 2, nothing, no green. So they were completely blocked. by the infused antibody, essentially proving the idea that this phenomenon actually occurs in people, and that putting in antibodies of these classes will make the immune system, in fact, target other regions. And that explains what happened in vaccinated people in terms of the breadth of their response and their ability to recall in such a way that even if they get infected with Omicron, these mutations, well, they're going to target down here and they're going to be okay. All right, so I've been droning on for quite a while, so I'm going to skip one more bit of data, which relates to the fourth vaccine dose or people that are infected after receiving 3 doses. Basically, the fourth dose doesn't do a lot. Right, so just to summarize, we've been following people at Rockefeller Hospital, trying to understand what happens in immunity to a new challenge, something that we really haven't been able to do before. We've used this tool, which is a way of finding cells that make antibodies of interest, to do most of what we've done. We've learned a lot about immune diversity, really, and the importance of that diversity in recall to variant viruses. In fact, we may have evolved this whole thing for this purpose, in order to be able to deal with the next virus. And in doing all of this cloning and learning about the response to this particular virus, has really been eye-opening. We're very, very fortunate with this virus that antibodies that target multiple regions really are effective. And that's quite different from other vaccines which are much harder, for example, HIV or influenza even. It's the same properties of the immune system, this diversification, this requirement for diversity, that actually interferes with our ability to create vaccines for those viruses. But in this case, we're very lucky that what we have in memory is really preventing what could be a much more serious set of events. All right, so this is a work of a lot of people, and in particular, the clinical team that's headed by Marina Kasky, who is a Brazilian physician at Rockefeller, professor at Rockefeller, has been instrumental in this set of studies and in many of all of the human studies that we've done. The BNASH lab is the virology lab that we collaborated with, and in addition to that lab, the Rice lab, another virology lab, Charlie's Nobel laureate from a year ago, and the Bjorkman lab for the structural studies. And I'm happy to take any questions if there are questions. So, we're just a few people here, so feel free.
Alguém tem alguma pergunta, Professor Michel?
And you could be in Portuguese, I'll understand it. It'll be okay.
I can start.
Okay.
The questions. It was a very amazing talk and trying to understand how these antibodies and how the different strains were from the virus and different ways that they vaccinate the people and how it works over time. One of the things that you mentioned at the end, that is the response, so if I understand that correctly, either the passive antibodies or if we have our own antibodies sort of blocking the old epitopes, so the response goes to other regions, right?
Yes.
So my first question is, why in the RBD the first response is going first to probably this top part as you showed, and it's not random how they go? Why they preferentially go to that top part?
I think that's just based on physical chemistry. The way this spike works is the receptor binding domain is at the very, very top, and it tends to flop up, and when it flops up, it shows its very top of the molecule, and so it's most accessible, and that's, I think, is probably the simplest explanation.
Just a matter of the biochemistry, and that's more available.
It's just the chemistry of it, and it's the availability of the antigen.
And that corresponds also to the IgA, as you're saying, in the membranes or in the airways. Do we see that change also in the IgA in the airways?
The IgA, we haven't studied the IgA response very much, but we did clone a bunch of IgA antibodies. They were initially similar to the IgG antibodies. The response that's in the airways is similar in many, many respects to the general response.
Okay. Yeah. Okay. So my final question is this. With the influenza virus that we usually get all the different strains every year, and we make a different vaccine based on what has here in Brazil, what has been more prevalent in the North Hemisphere in the previous season to get a better cover for the vaccine.
Yeah.
Probably for the coronavirus, we won't have this kind of cover because we don't know what different strains will eventually come up, right?
Well, this is very complicated. Yeah, so your question has many parts to it. First part is, why the heck do we need to make this every time, right? Why do we need a new influenza? Why can't we just have a universal vaccine for influenza? And the answer has, I believe, a lot to do with this blocking that I showed at the end there. So in influenza, there are some universal epitopes. There are really two of them. They're actually quite small, and unlike this coronavirus RBD, where anywhere you touch it, you make essentially a neutralizing response, you will not get a universal response to influenza unless you hit exactly there. Now, your previous exposures to influenza mean that you have antibodies that actually, they don't go right into that spot, but can interfere with it. So that you really, no matter how many times you're boosted, you're not going to get in there because you're blocked. That's the same for HIV. So that's one part of your question. That the targets are so specific and so covered up that antibodies that you're already making that are non-neutralizing are going to interfere with subsequent vaccination and a universal vaccine. The other part of your question is about what are we going to be able to predict about subsequent coronavirus infections. And I don't think we can predict very well at the moment. Part of that has to do with the way the virus is evolving. So the initial virus, the Wuhan, produced a series of variants, including the Brazilian variant, going up sort of this way, you can imagine, in a tree, in an evolutionary tree. And Omicron is something that came out this way. And subsequent evolution is proceeding through Omicron and not through Wuhan. We don't know if there's going to be something that comes off the tree going that way, right? So it's very, very difficult to predict. But the good news is that irrespective of this diversification, the Wuhan vaccine, the original vaccine, has been really good about protecting us from hospitalization, probably because of memory, of the diversity in memory. And it's continued, I think it'll continue to be quite good. So I think we're actually in amazing shape for this thing.
If we keep, as you showed, simulating new clones with subsequent vaccinations, vaccine shots, this might induce some sort of better cover to new variants as well, right?
Yeah, so I think that the third, I didn't get a chance to show the fourth vaccine dose, but the fourth vaccine dose does very, very little. Okay. It's really the third one that matters most. And maybe that's because the interval between the first and second was too short. It may have been that a second dose spaced further to allow some evolution before vaccination would have been just as effective. But fourth doses or fourth exposures do very, very little.
Eu não quero monopolizar, desculpa.
Hi, Michel. Thank you for your talk. So if you have a diversity of memory cells, in fact, you are protected for the variants. Have you seen difference between the methodology of the vaccines, like the mRNA or the adenovirus or the in the neutralizing antibodies and in the protection?
Yes. Basically, there are two classes of vaccines. There are vaccines that have a stabilized S and vaccines that do not. The vaccines that do not have a stabilized S include the adenovirus vaccines, the Chinese vaccine, the Russian vaccine, J&J, whatever, all of them basically, not stabilized. The other class, stabilized, is essentially the mRNA vaccines and now Novavax. So 3 that are commercial. The response to the non-stabilized is inferior, and it's significantly inferior. And the reason is very simple. When you put the non-stabilized protein into a person, it goes boom. And the targets for the immune system are no longer restricted to the important ones, but you have all kinds of junk. So you respond to junk, you make a little bit of the right stuff, but it's not as much of the right stuff as if when you have the stabilized form and get the real thing. So it's a pretty simple explanation, actually.
Mais alguma pergunta?
Okay. Very good. Thank you for your attention.
So thank you very much.